Thank you. That was a beautiful talk, and I think also a beautiful illustration of the point that Aviv made on the panel, that we have to extend beyond looking at cells and start looking at tissues and different cell-cell interactions and cell behavior in that context. The next speaker is Ollie Jonas. Ollie did his PhD in Germany and was actually a graduate in the physics department. He then conducted a postdoc with Bob Langer here at the Koch Institute and did a project in collaboration with the SEMA lab, where he developed microdevices that are implanted directly into tumors and have the ability to assess the optimal therapy for treatment by monitoring the response to release of many different drugs in a micro location. He started his independent position in 2016 at Harvard University in the Department of Radiology. He's now an associate professor there, and his laboratory is located in the radiology department at Brigham and Women's Hospital. The title of his talk is Lab-in-a-Tumor Microdevices for Multiple Drug Delivery Measurements in Cancer Patients. Please welcome Ollie.
Okay. So first of all, thank you to Matt and the organizers for having me here today. It's really a wonderful occasion to come back, and I have the absolute fondest memories of my time here. And I think among the speakers here, I may have one of the more unusual paths to get here. So I studied physics in grad school, and at the end of that, I basically wanted to get away from the lab and probably never see a lab from the inside again. And I ended up working as a management consultant in a completely sort of non-scientific industry. And through a sort of weird twist of fate, got introduced to a partner at a local venture capital firm here, Bob Tepper. And we started working on some of these ideas, started throwing around some crazy ideas about addressing some unmet needs in clinical oncology, and came up with a concept to maybe use an implantable device to actually measure drug responses in patients. And then we reached out to Bob Langer at the Koch Institute here. And I remember meeting with Bob, and we told him about the idea, and Bob loved it right away. And at the end of the meeting, Bob looks at me and says, "You should really be the one who makes this happen. You should come to my lab as a postdoc." You know, I was really impressed by what Bob had told me, what I had learned about the Koch, all the wonderful things, you know, bringing together scientists and engineers. Sounded amazing, but of course, being a postdoc has its sort of life challenges, the career uncertainty, the low pay. So I went home to my wife and told her that, you know, this sounds really amazing, but I know it's not the best time in life because we were expecting a child then, and it's like, okay, well, you know, nothing like starting a career with low pay and high uncertainty at that time. But she was fully supportive, and she said, "You should do this, it's amazing." You know, shortly thereafter started in Bob's lab, and the Koch to me is really not just sort of a personal you know, journey of taking a chance and having such an amazing experience, but also really sort of being a part of that coming together of engineers and cancer biologists. And I think the project that I'm presenting today, I think illustrates that very nicely. So as everyone here knows, one of the great challenges in clinical oncology is identifying which patients should receive which chemotherapy treatment. There are many drugs to choose from. They're often given in combinations. They're toxic. They cost a lot. These patients sometimes only have maybe 1 or 2 shots on goal. You know, there's certainly clinical guidelines, there's standards of care, and more recently through through genomic and other techniques, we have the ability to tailor therapy to some degree, but it's still the majority of patients really isn't benefiting tremendously yet. So that was sort of the first major unmet need. And a related unmet need we also found in drug development, where many of the drugs that end up failing, they fail late in a phase 3 study, and they often fail due to a lack of efficacy. And so we thought, okay, is there a way to maybe get some of this data much earlier before we invest all this time in PK, in tox? We go through phase 1, we go through phase 2, and then we find out in phase 3 the drug doesn't really work. So that's what sort of gave rise to this idea, okay, is there maybe a better way to get this data earlier? The response of cancer to drugs is really dictated by an incredibly complex interplay of the genome and the epigenome and the transcriptome and the metabolome, and all that contributes to the phenotype. And so the first sort of concept that we developed was, okay, this needs to happen, like any measurement we're going to make needs to happen within the tumor. So we can take all those factors that end up contributing to a drug phenotype, that we can take those into consideration. The question, of course, becomes, how do you do that? How can you do it for multiple drugs at once? Given the toxicology challenges, given the delivery challenges. And then we kind of came across this FDA concept of a phase 0 microdosing trial. And that was, at the time, was being used for PK studies, essentially. So a microdose was defined as less than 1% of a systemic dose, of a therapeutic dose. And people were doing these microdose injections to study how drugs distribute in the body. But our thought at the time was, okay, maybe we can actually deliver a microdose directly into the tumor. If it's a small enough region, then it can be a therapeutic dose locally, but systemically across the patient, it's still a microdose. And then there's a favorable regulatory process for that. And then the concept that we ended up coming up with is this implantable microdevice that's smaller than a grain of rice, and that's delivered directly into the tumor through a biopsy needle, and then remains in the tumor and can measure the response of the tumor to multiple drugs. So the technical workflow looks like this. So this is what these microdevices look like. Oops, I don't know if I have a good cursor here. Oh, yeah. So they're placed directly into the tumor. This is an example of a sarcoma here, and then these devices remain in the patient for multiple days. We now typically leave them in for about 3 to 7 days, but depending on the drugs, that can also be longer. And then the device is removed with surrounding tissue. That can either be a whole tumor excision or a coring biopsy around the device. And then you obtain these cross-sections of the tumor at each device level through sectioning, and then we can measure the actual responses of the tumor to each of the different drugs, typically about 20 drugs per device, but we can always place multiple devices in the same tumor. And so what we thought of as the key capabilities of this platform was, one, the high throughput, so being able to test many single agents, many combinations in the same tumor, in the same patient. It had to be minimal toxicity because we're giving so many different drugs. It had to be a very small local dose. We were hoping that this would allow us to measure a local PK/PD response to approved agents, to investigational agents, to novel combinations, and then to do all of that within the local tumor microenvironment, and to eventually to use that information to sort of tailor therapy for each specific patient. And so some of the first prototypes that we built then did not look as nice as this one. They looked much sort of less sophisticated, but with Bob and with Mike Sima together, we started making some of these first prototypes. We thought, okay, a great way to deliver this is through existing biopsy needles, and so that's where that cylindrical shape came from. And then there were some sort of engineering modifications along the way. We then had to figure out, okay, how are these best implanted into the mouse tumors? How can we retrieve them? and then how do we process them, section them histologically so that we can actually measure the drug effect. So that was sort of a big kind of step-by-step process of kind of developing this for the first time and testing it in animals. And of course, the drug delivery aspect was really critical to this, and that's where sort of Bob's expertise and many people in his lab were just so supportive and so helpful. So we ended up deciding on a matrix formulation, sort of polymer controlled delivery. These are some of the first sort of attempts at that are shown here. Using different polymers, the PEG polymer ended up being a great choice because it's easy to use clinically, and it also has a wide range of molecular weights, so we can really tune drug release to the specifics of the drug and also of the tumor tissue. And then a key sort of early aspect in the development was really tuning those local drug concentration from the microdose release so that we can match what reaches a tumor from systemic dosing, because obviously dose is an important part of efficacy, and so being able to match the intratumor dose to what you would get from systemic dosing is an important part, and some of that work is shown here. The detection of the drug, again, is key to sort of confirming that it was delivered accurately. Some of this work was done actually in collaboration with Sean, where we used mass spec. That's shown in an example here to detect some of these small molecule agents. We also have some drugs that are autofluorescent, so you can not only sort of theoretically calculate what polymer and how you formulate it, but then you also measure it directly to confirm that it was delivered in the quantities that were desired. And then we have a concentration gradient of how the drug is being released into the tumor. And that's an important part of the measurements that I'll elaborate on a little bit more. But it's basically the closer to the device, the higher the local concentration, and then the further out you go, the lower. And so you're able to sample a drug also across a range of concentrations and really get some of those PK/PD measurements within the tumor. So one of the first kind of major milestones of this work was about 3 years into my time here. So we were able to show that these local readouts from the microdevice can be used to predict systemic efficacy. This is an example of that, of a study that was done in triple-negative breast tumors. And we showed across a range of drugs that there's a one-to-one correlation between the device readout and the systemic sensitivity. But of course, the Langer Lab and the Koch Institute being what it is, we also started talking to some of the more tumor biology focus groups and started working with Sean and with Matt van der Heijden, and we wanted to use these microdevices to probe cancer metabolism in live tumors. And so one of the studies that we began talking about was related to macropinocytosis and protein catabolism in pancreatic tumors. And what we did here is we used our microdevices to deliver some fluorescent metabolic probes directly into the pancreatic tumors of mice. And then we were able to show that this process of protein catabolism happens in the tumor cells preferentially. And then we later on showed that the process by which that happens is macropinocytosis. And this was just an amazing experience, working with Sean and with Matt, but also at a technical level, being able to place the devices into mouse pancreatic tumors. If someone's ever done mouse work like that, the mouse pancreas is really small, it's really squishy, and you have to move a bunch of organs just to get there. And just on top of that, we then use 2-photon imaging on these live mice to actually observe those processes. So that wouldn't have been possible, I think, at any other institution, and I think would not have been possible without the amazing animal skills that Sean had, being able to do all this on live mice and keeping them alive and being able to do all these measurements. So that was a really incredible experience. In another study, we worked with the Frank Gertler lab, Madeleine Uden, and we used a similar approach using the microdevice to deliver fibronectin. And the idea here was to see if these metastatic breast cancer subtype called mean-invasive, if these cells would preferentially migrate along fibronectin gradients, and that's what's shown in the figure on the left here. The fibronectin gradient in red, and then in green we have this collagen network that forms along these gradients, and then the cells preferentially migrate along them. So that was kind of the first demonstration of this effect in vivo. Again, using the microdevice technology, but then combining it with some really interesting questions in cancer biology. So having done that, I think the next sort of challenge of the project was, okay, we've shown that this can work in mice, that we can do some really cool studies, but how do we do this in patients? And so some of the challenges, and this is not a comprehensive list, but this is sort of how we started thinking about this. Okay, so we've done this in mice, but doing it in patients is going to require clinical grade manufacturing. It's going to require formulating the drug in a way that's different from how this drug is being used clinically, and that sort of triggers a whole different set of regulations. Then there's the whole question of sterility and endotoxin burden. The FDA has pretty strict guidelines on maximum endotoxin levels, for example. So how do you actually make these devices in a way that conforms to all of those requirements? The question of regulatory path for this was basically completely unanswered. There was nothing exactly like it that the FDA had looked at previously. So one of the first questions was even, okay, is this a drug or a device? To us, it seemed like a device. To the FDA, it turns out it's more of a drug. So it's a combination product that's being regulated by the drug side, so by CDER. And of course, that was sort of a lengthy process just to even sort of establish that pathway. And then once we got there, okay, on the clinical side, how do we actually place and retrieve this in a patient? It's obviously a completely different challenge compared to the mice. There has to be image-guided placement for deeper tumors. It's a somewhat different procedure in each organ. There's different imaging modalities, for example. And then, of course, importantly, how do we integrate this with the existing clinical workflow? There's only so much you can do that's not part of the standard of care. These patients are, of course, we don't want to interrupt their treatment. So it's a pretty, I would say, major challenge just trying to figure out, okay, how can we integrate this into the clinical workflow? And then, of course, you need a team that has a medical oncologist, an interventional radiologist to place the devices, a surgeon who takes it back out, a pathologist who helps interpret the results. So it's a pretty major effort just to get over these hurdles. And what ended up happening is that we were able to work with the FDA and to establish a process that would allow us to do that, and that now I would say is a pretty smooth process that is typically only about 3 to 4 months. So we start with a clinical protocol. There's an IND submission. Then the FDA provides comments and concerns. There's usually a sort of feedback, and then resubmission, and then we get FDA approval, and then we go to the IRB at the local hospital. For most of our trials, that's Dana-Farber. And then the IRB, again, it's a bit of an iterative process, but once that's approved, then you can go into patients. And on the device preparation side, we had to work with the device side of FDA to really come up with protocols of how do we sterilize them, how do we make sure the drugs are not affected by the sterilization. That's also a sort of major concern. But fortunately, we were able to sort of work through that process and overcome those hurdles. And we currently have 6 clinical studies where the devices are being used. We have over 100 devices that have already been implanted and tested in patients across different cancer types, and have several more studies that are coming up in the near term. So I want to talk a little bit more about sort of the clinical implementation of how we're moving this forward in the next few slides. So the clinical procedures by necessity, we had to develop sort of based on the organ in which the device is being implanted. So we've developed a number of different ways to place and retrieve them. Some of those are surgical applications, so where the device is placed and then retrieved as part of a surgery. The interventional applications, where the placement and retrieval is done by biopsy, so by an image-guided biopsy. And then in skin tumors, we developed a slightly modified version of the device that's placed and then cored out by a dermatologist in the outpatient setting. This is an example of the implantations in some of the deeper lesions. This is a prostate patient here, where you see the lesion. Let's see, over here, and then you see 3 of these microdevices implanted. They're radio-opaque in the tumor, and then after excision, post-surgery, we confirm that the devices came out with a surgical specimen. In the skin, it's a little different process. They're shown on the bottom. The devices are placed by the dermatologist just visually, basically. You can see these lesions, so they're relatively easy to identify. And then you see here in the middle picture, you see 2 of these devices with little guide wires attached in the patient's skin, and they remain there for multiple days. And then we come back with a biopsy punch a few days later, and then take out the tumor specimen containing the device, and then that's processed in the lab, and we can measure the different drug effects. Of course, it wasn't the first device design that ended up making it to the clinic, but there were a few iterations, some changes that we made along the way, starting with the sort of cylindrical rod, and then making some changes that would reduce tissue damage, some changes that would improve the anchoring. We then added a guide wire later on. But what we found is in some of these patients, they actually, over the course of those 3 days where the device is implanted, they may accidentally sort of push on the device, or they may sit on it, or something else can happen that may dislodge the device. And so we ended up making a different design that looks a little more like a thumbtack, and sort of sits on top of the patient's skin. So there's always sort of modifications, and in some ways the engineering part never stops, even at this stage. One of the first implementations of this clinically was in lung cancer. So we placed the devices into different lesions, and then they came out with a surgical specimen. And that was really some of our first attempts at delivery and then measuring drug responses. We learned a lot from that trial, and then we're able to get some of these drug phenotypes that I described earlier directly from the patients, are able to understand how drug responses can change across different agents. But we quickly realized that one of the really important aspects is being able to use this, for example, in metastases. Some of these primary tumor patients do not get therapy after surgery, and so the probably highest unmet need is in the metastatic setting, so being able to place and retrieve the device using a biopsy. And so Sharath Bhagavatula, who's an interventional radiologist in my group, a staff physician, but has part of his time that he spends on research in my lab, is building some of these new devices, new biopsy tools that are able to retrieve the microdevices selectively from deeper tumors. So that's one of the areas that we're working on now. One part that I'm really excited about is the sort of biomarker discovery piece, and being able to test new combinations of treatments, novel agents entirely, in these patients. And so what's really changed, I would say tremendously over the last 5 years or so, has been the ability to use these spatial omic techniques, and Sean has talked about some of them. But they've really greatly expanded our ability to do deep phenotyping on these drug effects and really learn a lot more about drug responses than only looking at apoptosis or proliferation, for example. So this is some examples from our clinical studies, for example, using cyclical immunofluorescence, where you can do a deep phenotyping of the immune responses to each drug, doing spatial transcriptomics, or the MALDI mass spec imaging that Sean described. So in terms of the biomarker discovery, I would say we sort of group the readouts into two categories. The first one is more the functional readouts, so like a direct sort of PD readout of how much drug do I need to reach a certain level of target inhibition. That's an example here of enzalutamide and the androgen receptor from a prostate patient. Looking at the concentration dependence of different drugs, so looking at efficacy using a low concentration versus a high concentration, and then looking at combination effects. So this is again a chart here from a single patient looking at nivolumab as a single agent, but then also in combination with multiple other agents. So being able to do these sort of N of 1 studies for many drugs in a single patient, I think at a functional level, can hopefully yield some clinical insight there. But also on the biomarker discovery, using spatial transcriptomics and being able to sort of do a deep phenotyping for all 20 of the different drug responses that we're able to measure in a patient, and then doing a lot of comparisons, up to 120 comparisons for each device from different drugs. So one of the areas that we're pursuing now is in the combination of targeted agents and immunotherapies. That's, I think, a great unmet need, but also great potential there to optimize therapy. And we identified as part of this prostate trial a nivolumab olaparib combination that seems to be very favorable there, and starting to identify some of these pathways that are activated and that can be leveraged with the immunotherapy in response to olaparib. One of the really exciting things that are happening in our lab now are led by Zuzana Todorova, who has been working on trying to predict combinations between targeted agents and immunotherapies using the microdevice. So what Zuzana was able to show is that we can use the microdevice to deliver agents locally into the tumor, and then she combined that with a very sophisticated spatial analysis profiling of the immune response to the drug effect, and then spatial analysis of the different interactions between those local responses. And she was able to show that based on that local phenotype, we can predict the specific immunotherapy that would be most effective in response. And then she showed across a range of systemic studies in mice that is actually a superior therapeutic strategy, and that this local microdevice response can predict the best combination treatment. And the last thing I want to show is sort of, it gets back to the question we started with. Is there a way we can use this in patients to predict who's going to respond to a given therapy, or what the best therapy would be in a given patient? So glioblastoma is an extremely difficult to treat disease. These patients typically have about 15 months of survival. There aren't very many therapeutic options. The last drug approval in this disease was 20 years ago, temozolomide. And there are very few markers that allow us to predict who will respond to what. And since one of our studies is in glioblastoma, we were able to, for some of these patients, actually see if the device readout would have predicted the response to temozolomide. And so we were able to show for 3 patients that actually received that therapy, that the device predicted perfectly who will be a responder and who will not be a responder. And the interesting part about that is that for 2 of these 3 patients, the MGMT promoter methylation status, which is the only clinically validated predictive marker for temozolomide sensitivity, would have predicted this wrongly. And the microdevice measurement actually predicted it correctly. And so we think this is, even though it's early data, it's only 6 patients and only 3 of them actually received treatment. We think this is very encouraging, and we're excited to sort of take this work forward. I think in terms of looking ahead, I think there's 3 sort of major applications that I hope to continue to work on and that our team will continue to work on. I think that personalized therapy selection is maybe the first and most obvious one. I think on the drug development side, I think there's tremendous potential to use this to get drug phenotypes while the drug is still in development. And we actually recently worked out a path with the FDA to use non-approved compounds and even compounds that have never been tested in patients before with a very minimal regulatory burden. So I think there's tremendous potential there in testing new therapies. And then the last thing is really kind of putting all of that together, all the genotyping, all the multi-omic data that we're collecting with patients, and then connecting that with actual drug phenotypes. And we get so many drug phenotypes from a single patient that I think building those genotype-phenotype correlations, I think, could be a great way forward here. So with that, I'd like to thank our really amazing team of clinical collaborators from all across the Brigham and Dana-Farber and MGH, our scientific collaborators, many of which are here today. It's been an incredible privilege to work with you, and then the Jonas Lab, especially Susanna, Sharath, and Christine, who've contributed to a lot of the work that I showed today.